Hi, welcome to Social Research Insights. The mission of Social Research Insights is to promote open source software for data analysis, for both academic and corporate research needs. This is in fact one of the parts of my series of presentations on bioinformatics through R language. In my previous video I've explained how to compute GC content through R editor. I also explained what is the meaning of GC content and different aspects related to that concept, along with the ATC to GC ratio, etc. And this is the other part in continuation to the previous video. And in this part I'm going to explain you the gene sequence analysis. So we have pretty few methods to perform analysis on genome. They are gene sequencing, gene expression, and microdata array. So these are the three important aspects as long as the data analysis for bioinformatics is concerned. So there may be many things in genomics, but the purpose of these videos is to explain the data analysis. Given some data file, maybe in the form of a nucleotide or a genome, how do we need to do the data analysis? So that is the crux of my presentations. So in this video, as I stated earlier, before, just a minute ahead, I'm going to explain you the gene sequence analysis. So what does it mean, gene sequence? So in any given genome, when we download the data file from a particular genome bank, it is actually a list object, as I have explained to you in my previous video. It will be having so many elements, as in levels. So the gene sequence is one of the elements of that list object, which has the sequence of the nucleotides like adenine, thymine, guanine, cytosine, etc., etc., etc. So what exactly is gene sequence, and how do we try to understand gene sequence? So that is the purpose of this present demonstration. I'm not going to throw much light on gene sequence methods. In genomics, there are precisely well-defined methods to find the gene sequences. The scientists are very much busy in coding the sequence of different species. But the purpose of this demonstration is not to explain the methods. However, I'll just put very little. I'm just going to throw very little light on these methods. I'm going to provide the summary of these methods through this presentation. The gene sequencing methods, there are dozens and dozens of methods, and scientific community is still very busy in identifying and improving the methods. They can be summarized in four different categories. That is the category number one, the first generation methods. We simply call it as first gen methods, and we have wonderful some of the methods like Maxim Gilbert sequencing actually comes under this method. Maxim Gilbert I think Maxim Gilbert sequencing comes under. And next one, these are the two most important methods under the first generation method. Chain termination method. The another one is chain termination method. These are the first, and there may be a couple of other methods under first generation methods, but these are regarded as a few of the important methods under first generation. The the chain termination method is also known as the Frederick-Sanger method. Sanger perhaps is the father of gene sequencing methodology. Is known as I don't know. He is one of the important individuals in this area of study, because he has triggered the momentum for the advanced methods that we are using today in our labs. So mostly the bioinformaticist talks about the Sanger methodology while mentioning about the gene sequencing. So we have the second generation methods, sex generation methods, in which we have some certain wonderful methods like shotgun method. We have shotgun method is actually invented by Craig Venter while he was struggling with in a way finding the full genome for Homo sapiens, I mean human species, but they could do only part of the genome, but they had done some wonderful experiments on different species. They use a shotgun method to figure out the full genome of some certain species. And the next one is the other method is PCR method. PCR is the short form for the polymerase chain reaction method for gene sequencing. Polymerase chain reaction method. This is the other end of the chain termination method. This is chain reaction method. The chain termination actually belongs to the first generation methods, and chain reaction methods belong to the second generation methods. And we have the other methods, group of methods or category called commercial methods. Few firms tried in inventing few methods. One of the methods that we talk today is the Polony sequencing. It is actually a commercial method which originally invented by the Shendine and Poreka, whatever it may be. Okay, I'm not worried about the people who invented this, but this is regarded. Polony method is one of the commercial methods, and the other method is the solid sequencing, and these are also the very advanced methods known as advanced methods. The extensions of these methods are under study, and they are evolving. And we also have the next generation methods, and there are so many. It is very difficult to list out the number of methods that falls under the next generation methods. Under next generation methods, there are few types of methods that are pyrosequencing, Polony sequencing, solid sequencing, chain termination methods, and single molecular real-time sequencing, SMRT. And also there are methods known as massively parallel signature sequencing, method, and the other one is single molecular real-time sequencing, and solid sequencing, and also Polony pyro ion semiconductor method, single molecular real-time sequencing. The 454 pyro sequencing, Illumina. There are many things which we can discuss. The idea of this demonstration is not to cover the methods of sequencing, but to throw some light on the data analysis available for uh, gene sequencing. Okay, so but I just would like to cover some knowledge on the availability of different methods for gene sequences. So scientific community use few of these methods in order to figure out the gene sequence, which is one of the essential parts of the genome of any given species. So the data analyst job starts once he or she acquires this gene sequence from the data banks or the repositories like NCBI, EMBL, DDBJ, and etc. There are quite a few gene banks from where we can retrieve the data sets. And once the dataset, those datasets are manufactured by this scientific community through these methods. But once the dataset is available from the repositories, then the job of the data analyst starts, and we have pretty clearly mentioned statistical methods to do the analysis on the gene sequences. And this presentation, the idea of this presentation is to throw some light on these methods, I mean the statistical data analysis. This is some introduction about the gene sequence. There is some small description as why the gene sequence analysis is required. The purpose of the gene sequence analysis is to understand the biology or the gene product of a given species, and it actually involves using analytical methods on DNA and RNA to understand the features, function, structure, and also the level of evolution of a given species. And there is also another important observation in the scientific community that mere correlation of the genomes is not the job of the genomics, but the active understanding of those genomes and augmentation of the ecosystem is the main objective of genomics. So just having the data is not important, but we should be able to understand that data and take certain important decisions by understanding the species or organisms, so that will benefit the development, the safe survival of the nature. Okay, and this is in precise, like the very short formula for if somebody asks me what is gene sequence analysis, I just simply put it in this way: gene sequence analysis is equal to alignment plus comparison. So what exactly we are going to do there? There are certain important functions of the gene sequence analysis. I mean, the idea of gene sequence analysis is to gain knowledge on biology through some certain concepts known as similarity, which means we can take any two different genes and we can compare them and try and can understand the similarity between those two genomes. And we can also understand, for example, if we have the genome of banana and genome of apple, so we can study the similarity of the gene and try to figure out the differences, or the dissimilarities, and the why a particular fruit is different from the other fruit, and where exactly it is different, and why it has a different aroma, different chemicals inside. So that helps us to, that is actually the job of the homogeneity, a special branch of statistical analysis. We have pretty well defined methods to compute the homogeneity in statistical theory. So that we will discuss later, like what are the different measures or the methods or techniques that we use in order to compute the homogeneity between any two given genomes. And there is another thing, the genomic analysis is also helpful in order to understand the intrinsic features like the structure, in terms of the introns, exons, and active segments of the gene, etc. And the variations, we can also compare the variations. We call it as a heterogeneity, which is the opposite idea of homogeneity. We have very wonderful and well-defined methods to compare to assess the heterogeneity among the genes. That way we can understand the dissimilarities as how the genes are different from each other, where it happened to be different, and in which way we got to understand those differences in order to gain nice insights on the organism. And genetic diversity, so the universe is highly diverse. I mean, the nature, the diversity is the property of the nature, and where we try to understand the nature through this idea called diversity. And to do that, we need genetic information. We can analyze the genetic data, the genomic information of a particular species, or the different types of species in the nature, and we'll also try to understand the diversity. And identification of the molecular structures, which will help us to gain a very in-depth understanding of a particular species or organism. And these are the two important aspects in sequence analysis. We call them as the sequence alignment is one of the important component in gene sequence analysis. And there are two types of analytical methods of sequence alignment: pairwise alignment and multiple alignment. This is what is important for me to explain, because the idea of statistics or data analysis comes into the picture when through the sequence alignment in gene sequence analysis. So this is rather very important for the data analyst. So we have very good packages and as well as the techniques in this body of knowledge to understand the gene sequence. To do analysis on the gene sequence in order to gain the understanding on organisms. So these are the two important methods that we employ. All those techniques falls under these two categories, that is pairwise alignment and multiple alignment. All those techniques of sequence alignment falls under these two categories, that is pairwise alignment and multiple alignment. We have wonderful packages as well as the methods, well-defined methods in order to do data analysis related to this particular aspect of genomics. And what is the entire idea of gene sequence analysis? I just brought within a paragraph. The common use of the pairwise sequence analysis is to take the sequence of interest and compare it to all known sequences in a database to identify the homologous sequences. In general, the matches in the database are ordered to show the most closely related sequences first allowed by the sequences with diminishing similarity. These matches are usually reported with a measure of statistical significance, such as the expectation value. This we'll try to discuss what is this expectation value in statistical methods, but this is the idea of the pairwise alignment. We try to understand the various segments of the sequence, and later we pull different sequences, the sequences related to different species, and try to find the similarities among these segments, and try to do some statistical statistical analysis, and finally we try to understand the organism. So for example, if we are studying about a particular virus, and we can pull the information about this virus in different gen banks, and we can just obtain the segments of this virus and try to find the similarities, and finally try to understand the nature of the virus based on the analysis which is performed through this method. Okay, that is the pairwise alignment analysis in gene sequence analysis. And these are the other methods. They are very much important while studying the gene sequences. They are called as profile comparison and sequence assembly, gene prediction, protein structure prediction. I can say these are the applications rather than the methods. The profile comparison is actually we compare the profiles in order to understand the characteristics of the species or organisms but sequence assembly is a special task of writing the sequence based on our understanding on the sequences on the fragments that we had studied through the sequence submitted by the scientific community. And gene prediction is the predictability we have wonderful statistical techniques to predict some certain important aspects of genes that through regression, and regression itself has so many versions like logistic regression, probit methods, and simple linear regression, multi variate regression analysis. There are many things, like we use all those statistical knowledge or techniques in order to improve the predictability of a particular characteristic through genomic analysis. And we have protein structure prediction. This is a rather complicated part of the analysis where the analyst will try to plot the 3D structures of the proteins and try to understand the different regions of the gene through this method. And these are the different statistical methods. Which perhaps I need to talk more and more on these methods because I am a statistician. We use all these methods in order to understand the genomic information of any given species. We have clustering, vector analysis, Markov chain models, artificial neural networks, and Bayesian network analysis, regression, and sequence mining. So in which we have this regression analysis, Bayesian methods, clustering, Markov chains, and artificial neural networks are core statistical methods, and rest of them are hybrid, like the statistics used used combinedly with the methods of genomics. Like when we talk about the DNA patents, it is actually the hybrid method where we use the methods that belong to both statistics and genomics. In the same fashion, we have sequence mining. This is also requires the knowledge of the bioinformatics and statistics to perform the analysis. But the other methods like artificial neural networks, Markov chain models, clustering, Bayesian statistics, these are all the very native concepts of statistical theory. Okay, so this is what my excerpt on gene sequence analysis, and in my next video I will try to explain how to perform data analysis on gene sequences of different organisms through our language. Thank you.